Welcome to Click for Biology. We're in the topic of plant nutrition, photosynthesis, and we're looking at experiments that demonstrate the production of oxygen. If you'd like a copy of this presentation, there's a PDF in the description link below. The demonstration of the production of oxygen is actually with plants which grow typically in water. We call these aquatic plant species. And of course, it's necessary for photosynthesis to provide the plant with a source of light, probably a bench lamp. And then we need to be able to collect the oxygen and then carry out a test to demonstrate that it is indeed oxygen. And the way we do this is to position the plant at the bottom of a beaker of water. That water contains hydrogen carbonate. Now, the hydrogen carbonate will dissolve in the water to leave carbon dioxide dissolved in that water. And of course, that's part of the equation for photosynthesis, that we have carbon dioxide able to combine with water in the presence of light and chlorophyll to create glucose and oxygen. And today it's the oxygen that we're attempting to demonstrate. So the plant is in water, it's provided with sufficient carbon dioxide, and as it photosynthesizes, the oxygen gas is given off from the leaf structures. This rises through the water, and we would start this experiment with a test tube or a boiling tube completely full of water. The oxygen which accumulates at the top of this boiling tube will displace the water downward. This is sometimes called collecting a gas over water, and another phrase for that is the displacement of water by a gas. Either phrase will work in this situation. So the inverted test tube initially is full of water, but as the oxygen bubbles come off, we can collect those, and then we would go on to demonstrate, and this is the critical part of the test, is to then collect the oxygen gas and carry out a test that demonstrates that it is actually the gas oxygen. And this would be the classic splint, the wooden taper splint, and it will first be lit, and then we will put the flame out. It will be glowing, and then in oxygen it will relight. So we say it relights a glowing splint to demonstrate that it's oxygen. So that's the full method for demonstrating that oxygen is given off during photosynthesis. It's important that students pay attention to this particular experimental setup, as this is often used in photosynthesis experiments and modified in various ways depending upon the intentions of that experiment. Okay, here's some quick questions to do. Probably take 3 minutes. Pause the video, do the questions, write down your answers. It's a really good way to improve your learning. So write down your answers and then go down to the description below, where you will find that I've posted in the answers so that you can check your understanding, and then move on to the next experiment, which is linked as the next video. There's a link to the playlist, and of course the chance to subscribe to the channel and maybe give us a thumbs up for the video. Thank you for listening.